Okay, thank you. So thank you so much for inviting me. So I'm happy to meet you and to discuss with you today. So Hervé told me that you were interested in trying to bring several disciplines all together, and so he asked me to give a talk on based on my experience. So I will present you what I understand about doing research at the interface between several disciplines. And in my case, it was developmental biology, genetics, evolution, and ecology. So I will present you my presentation will be divided into three parts. So first, I will present you my background and my work so that you can understand better why I'm talking to you today and why I have any suggestion and important points to suggest. Then I will focus on more general points about first the researcher and the research done by the researcher. And then I will talk about all the other things related to research, like publications, research institutions, funding, and a relationship with colleagues when we are working at the interface between several disciplines. So in my lab at Institut Jacques Monod in Paris, our goal is to try to understand the mechanisms underlying evolution and diversity in morphology, physiology, and behavior that we observe between animal forms. And I did my PhD in developmental biology, but now in the lab, our main discipline is evolutionary genetics. And we also interact with many other fields in biology, so physiology and metabolism, but also biochemistry, bioadhesion, and also ecology. And so just to go into more detail, so I did my PhD at École Normale Supérieure, and I work on developmental biology. And I was working using Drosophila melanogaster as a model system and trying to understand how different cell types are formed during development from a single cell and how cell differentiation occurs. So it was pure developmental biology, but I have always been interested in evolution. And at the end of my PhD, I started to address evolutionary questions. And then I really went into it during my postdoc in Princeton University with David Stern. And here I was interested in microevolution, so comparing closely related species which can still hybridize in the laboratory. So here I show you a picture of a human and chimpanzee, but I was working still with Drosophila species, closely related species of Drosophila, and trying to identify the mutations which occur during evolution and which contribute to phenotypic variation to try to better understand the mechanisms underlying evolution. Then I did a short postdoc in Japan, again on developmental biology and on my PhD work. And then I obtained a position at the CNRS in Jussieu. And here my project was on evolutionary genetics, so similar to my postdoc. And I was also interested in different types of phenotypes. And these types of phenotype allowed me to work at the interface with other disciplines, so physiology, metabolism, and again, developmental biology. And now, I have a group at Institut Jacques Monod, which started in 2010. And so, as I told you, so I mainly work on evolutionary genetics, but these days we are working on the glue produced by Drosophila species. So we work on bioadhesion, and we try to relate our project also to ecological questions. So throughout my career, I have always been working with Drosophila. So first with Drosophila melanogaster, and then in my postdoc with many species of Drosophila. And now we are always working with Drosophila and many species of Drosophila. I have been also interested in making a database, like summary of many data accumulated by many different groups. And so during my PhD, I created a database on the embryonic and larval peripheral nervous system, so describing all the different cells, the different antibody stainings and in insert trap and reporter lines and Galford lines that we can use to identify all these neurons and study them and how they were formed during development in melanogaster. And then for my postdoc, I created a database on the phenotypic differences that have been reported in the literature between species of the melanogaster subgroup. So you can see them here. And so morphological differences, physiological differences, and behavioral differences. And I mean, this is helpful, I mean, for people who want to study these species and want to identify genes underlying these differences. And then later on, I mean, with David Stern and then Arnaud Martin, we created a database called GFIBase, which is still ongoing and updated regularly. So it's a database of the genes and mutations which have been identified as contributing to a phenotypic variation, either on morphology, physiology, or behavior in all animals and all plants. And we also include yeast. So all we're trying to include all eukaryotes. So it's a big database. We have more than 3,000 papers curated in this database. And so it's a lot of work, but we are trying to maintain it because it can help to find general data about the genes underlying evolution. So this is for general work. So you can see here with the database that already, I mean, we are working not only with Drosophila, but with any animals and any plants. So we have to learn about these different model systems that are used by different labs. And when the pandemic started in 2020 in France, I mean, I was scared, very scared. And then with other friends, I mean, we decided to create a website, which is called adioscorona.org, to give advice to people. So first it was made for France and for French people, but then we translated it in many languages. So it's now available in 9 languages, and there are more than 150 questions and answers. So it was, the goal was to help people to know what to do during the pandemic, and also to give references to scientific articles and to give the reasoning underlying why do we have to put masks. I mean, what are the data? What should we do if we want to play sport or things like that? I mean, just basic questions, but with advice based on scientific literature. I'm also involved in research on a new technology called Gene Drive, which I will not develop today. So this technology was developed by mainly molecular biologists, but it's the main target of this technology is to regulate and to modify pest populations in the wild. So it has ecological aspects. And so I think this technique is quite dangerous, and there are many risks which should be studied while this technology is being developed in research labs. And so I've been collaborating here with a philosopher, Baptiste Morisot, and a biologist, Christophe Butt, on trying to see what would be the possible application of this technology. And then thinking also about the risk and how can we make sure, I mean, can we control in a way this technology, so using modeling and thinking about the various risks. And recently, I mean, I've been also involved in the origin of SARS-CoV-2. I mean, the hypothesis that it could be a lab accident has not been fully rejected. So it's still a hypothesis which should be investigated. And so far, unfortunately, it hasn't been investigating as much as it could be. So with other researchers, we published a paper on this topic, and I'm also trying to do a few epidemiological studies on, based on the data we have, to try to learn more about how it started. So these projects are more about the link between biology and society, so especially Adios Corona, and which is really close to the public. And then these projects on Gene Drive and COVID-19 origin are really projects which are based on data which are published by other people, and just looking at this data and looking at what we can conclude from the data. So there is no experimental work on this project. And last year, I mean, I have been asked to be part of the CNRS ethics committee. And so I invite you to go to the website. And on this website, you can see different avis which have been published, so all in French. And so it's really interesting, I mean, to learn more about links between researchers and society. And this year we are working also on something about how researchers interact with the public, and thinking about the ethics of this, because more and more researchers are asked to talk to the society and how this could be done in the best way. So this is about my background, general background, and my general work. So before going into what researchers do and how, I mean, what is best in my mind to work at the interface between several disciplines, I mean, I will tell you a bit more about research which have been done in my lab. So, for example, one research project we work on was to try to understand how a Drosophila species called Drosophila pacea adapted to a specific cactus. So this cactus is called the Senita cactus. It is found in Mexico, and this Drosophila species is found only on this Senita cactus. You cannot find it in other places. So we went to Mexico, I mean, to collect this fly and to try to understand how it adapted to this cactus. And it was known based on previous studies in the 70s that this species cannot be raised on normal fly food in the lab, whereas Drosophila melanogaster, I mean, they can be raised on normal fly food which contains traces of cholesterol. In this case, Drosophila pacea cannot survive on normal fly food. However, if pieces of the Senita cactus are added to the fly food, then they can survive. You can get larvae and adults, and you can raise them for several generations. And this cactus produce specific sterols, which are not cholesterol. And if we add a specific sterol close to the one produced by the cactus, latosterol, to normal fly food, then we can get flies for many generations. And in the lab, I mean, we don't use latosterol because it's a bit expensive. So we use 7-dehydrocholesterol, and also on 7-dehydrocholesterol, we can raise the flies, and we have been raising them now for more than 7 years in the lab by just adding 7-dehydrocholesterol. And in 2006, it was found that the synthesis of steroid hormone in Drosophila involves a first step, which is the conversion of cholesterol into 7-dehydrocholesterol. And that this first step is catalyzed by an enzyme called Neverland, or here NVD. And what is special with insects is that they are unable to produce cholesterol de novo. So they rely on dietary sterols. So they use a sterol present in normal fly food. And so this enzyme converts cholesterol into 7-dehydrocholesterol, and this is the first step in the biosynthesis of steroid hormone. So here, when we wanted to understand this specific metabolism found in Drosophila pacea, we had a good candidate gene, which was published in 2006. And we thought, okay, I mean, are there any mutation in these genes that would explain that there was a loss of this activity in Drosophila pacea? So we sequenced the gene in Drosophila pacea, and indeed we found several amino acid changes, which, but the enzyme is still present and still produced. And this enzyme with these 3 amino acids is now unable to convert cholesterol into 7-dehydrocholesterol. So we did several in vitro assays to test the action of these specific amino acids. And to our surprise, we found that this enzyme converts, is a switch substrate, and now converts latosterol into 7-dehydrocholesterol. And actually, it's not really a switch of substrate, because the Neverland enzyme from melanogaster and from many other insect species we tested can convert both cholesterol and latosterol. So what happened during evolution of Drosophila pacea is that several mutations accumulated in the coding region of the Neverland gene, so that the enzyme encoded by the gene lost the activity on cholesterol, but kept the activity on latosterol. And actually, this is a reaction which is used in Drosophila pacea to use the sterol produced by the cactus. And what we did also is that we put the Neverland gene from pacea into melanogaster, into a mutant for the Neverland gene in melanogaster. And what we could find is that these flies, they survive better on the cactus sterols than when they have their endogenous Neverland gene. So actually this mutation, they probably had an adaptive role, and they gave a gain of fitness in terms of survival on latosterol, even though you have a loss of activity on cholesterol, and actually the activity on latosterol is decreased compared to the ancestral activity or the activity in melanogaster. And so then we did also analysis of genomic sequences, and we found that there are traces of ancient selective sweep near the Neverland locus. So we have arguments based on fitness experiment in the lab, and also traces of a selective sweep to say that these mutations were selected during evolution of Drosophila pacea while it adapted to this cactus. So by doing this work, so which combined experiments using physiology, metabolism, and in vitro experiment with enzymatic activity, we could identify the exact mutation which occurred during evolution of Drosophila pacea. And this was really important because it changed our previous hypothesis that we had when we started to work on this topic. Because our first hypothesis was that Drosophila pacea first adapted to the cactus. And because the cactus did not produce cholesterol, the loss of activity on cholesterol was just a neutral loss, and there was no specific effect associated with this mutation. They were just lost because there was no function, the function was no longer required. But then, because we identified the mutation, we could see that actually these mutations, so then not only give rise to the loss of activity on cholesterol, but they also increase the fitness on latosterol. So they were probably selected during evolution as the fly adapted to the cactus. And so as a direct consequence, the fly became dependent on this cactus because it was no longer possible for these flies to use other types of plants which produce cholesterol. So here, this adaptation to the food source, gave rise to a metabolic dependence and to these flies, which are now pure ecological specialists and can only live on this Seminita cactus and on no other plant in the desert where they live. So it was really useful, to combine different disciplines to have a good understanding of these mutations and their role in the physiology of the fly. And so this was nice because we could publish in Science our results. And what I want to tell you about this also is that when I started at the CNRI, so I applied for ATIP-Avenir, and my project was on this project because I was working in a lab which was involved in steroid hormone physiology, and it was known for a long time that Drosophila pacea had a specific metabolism, and a lot of people working on a steroid hormone production in Drosophila were curious about these flies, but they were not into evolution, so they were not going into working on this. And so I thought it was a nice project to work on. And so I wrote a grant proposal, but it was rejected on the basis that I was not a physiologist and it was too far away from my expertise. But I was really happy that I continued. It was good that I continued because it allowed us to publish a really nice paper, and I'm quite proud of what we did. And I managed to do it with others, and it was really important to collaborate with other researchers, especially physiologists, for all the techniques and approaches that we developed to do the in vitro assays and test the role of the different mutations, and also with the evolutionary biologists. So we collaborated with Andy Clark, who helped us a lot also to test for a selective sweep in the region. So this is just an example, but this is also to tell you that it can be difficult, I mean, to work on a topic which is not the one we are known for, because grant proposals are usually rejected, but it's good to feel that we can do it and continue. And I mean, I always think about this example, and when now I have grant proposals which are rejected, I think, okay, I mean, it's rejected, but it doesn't mean it's a bad project, because I have this example which hasn't been funded, but still gave rise to a nice paper. Another example I would like to tell you about is a study, I mean, comparing Drosophila centomere with its closely related species, Drosophila jacoba. And so Drosophila centomere lives in Africa. And what is interesting is that on the genitalia, they lost a pair of bristles. And these bristles are highly conserved. They are found in all closely related species, but they are absent in Drosophila centomere. So here we use a lot of genetics, because we can cross Drosophila centomere with its closely related species in the lab. It's difficult, but possible. Whereas in nature, they usually don't hybridize. And so doing a lot of crosses and a lot of genetics, we identified one main gene involved in this loss of bristle, which is a gene encoding a transcription factor, SCUT. And we also found a specific cis regulatory element in which a T is mutating into a G. And this leads to a decrease in expression of the gene in the developing genitalia and a loss of bristles. And then we compared the bristle pattern in Drosophila centomere with for other parts of the body with its closely related species. And we found that on the first pair of leg in the male, there is a gain of bristles. And actually this gain of bristle is also caused in part by this same change. And here, the same mutation increased the expression of SCUT in this developing tissue, whereas here it decreased the expression. So here we have a case of a single mutation which affect two different organs. And so this single mutation which occurred during evolution affected two organs at the same time. What is interesting is that these two organs are involved in copulation. So here it's on the genitalia, and here it's on the sex comb. And the sex comb is a structure which is used by the male during copulation to grip the female. And so it's an interesting case where we have a single mutation affecting two different organs. And so it shows that there are several constraints during evolution, and such pleiotropic sites usually are unlikely to evolve, because usually you will have a deleterious mutation. I mean, some of the effects will be deleterious, but here in this case, we can see that it evolved, and it is found in all the Drosophila centomere flies that we sequenced, and we sequenced many of them. So it seems that it's fixed in the species. And so it's interesting that it modifies at least two different organs. And so this project was possible because we mixed developmental biology approaches with the evolutionary genetics approach. And the question we had was, how can we explain a recent change in a bristle pattern in one species compared to the other? And does it correlate with changes in bristle pattern in other parts of the body? So the question is interesting from the point of view of evolutionary biology, and also from the point of view of developmental biology. So now in the lab, the main project we work on is fly glue. So during the life cycle of Drosophila at the late third instar larval stage, the larvae are wandering around. I mean, they walk out of the food, and they start to find a place where they will pupate. And so within the pupae will occur metamorphosis, which lasts several days. And at the end of metamorphosis, the operculum here will open and the adult will emerge, and you will have a male and female emerging from the pupae. And this pupae is attached, as you can see here, with a glue. And this glue is produced by the salivary gland here by third instar larvae just before entering into the pupal stage. And this glue is made of several proteins. about 8 major proteins are found in this glue. And these glues allows the animal to attach to a substrate for several days. The substrate can be fruit skin. It could be also the wood or stones. And usually the pupae, so they attach to plants, or they attach also into the soil. And so here we want to understand how this glue evolved between different species that are adapted to different environments. And so we work at different scales. So at the genomic scale, at the level of the sequences of the glue genes, we try to understand also how these glue have specific adhesion properties and what is the chemistry of these different proteins. So we use chemistry and molecular biology approaches. We use also different approaches to study the adhesion of these pupae. And we also try to relate the adhesive properties that we identify in the lab to the ecology of the flies. So we work with many collaborators on all these different topics. So Jean-Luc Dallage and Rob Culatinal help us to work on the analysis of the sequence of the genes and comparing the sequence between different Drosophila species. We are also working with Yann Gerardel and Jean-Michel Camadro for the molecular biology and the chemistry of this glue. And for the adhesion part, we developed a specific adhesive test with the help of Stanislav Gorb and Alexander Kovalev in Kiel. And this was really helpful to design and develop this test. And so now it's good. We have a test to measure the adhesion of many Drosophila species. And this analysis is done in collaboration with Laurent Corté and François Graner, who are physicists. And we also work on the ecology. And for example, we published a paper where we found that this glue is important to protect the animal from predation from ants. And so this was done in collaboration with Mathieu Mollet, who work on different ant species in Jussieu. And so this work is done in the lab by different people. So Flora Born, who was a PhD in the lab for 3 years, and now she's a postdoc in Peter Randolph-Feather's group in New York. And now the main person working on the glue project is Manon Monnier, and she's a second-year PhD student. And she is co-supervised by François Graner, a physicist, and by me on this. And so she's analyzing a lot of different Drosophila species, characterizing the adhesion properties of various species, and trying to relate it to the glue genes. And then she's helped by Isabelle Nuez, who is a technician in the lab. And in the lab, we have also 3 other people: Alexis Lalouette, who is and Michael Lang, who are working on the evolution of genitalia in Drosophila, and Jean-Noël Lorenzi, who is working on the origin of SARS-CoV-2 and who is analyzing available sequences of SARS-CoV-2 and closely related viruses. So this is for a general view of my background and the work we do in the lab. So now what can I say about the researcher and the research done by the researcher when we are working at different disciplines? So I went back to this paper, which was published in 2006, where they compare multidisciplinary, interdisciplinary, and transdisciplinary. And they say that you have different steps in the way that different disciplines can be mixed. So multidisciplinary means that you have combined knowledge from different disciplines, but each discipline stays within its boundaries. Interdisciplinary is mixing a little bit more. So here you have a synthesis of several disciplines into a coordinated and a coherent whole. And then transdisciplinary is like a next step, where the different disciplines are integrated, and then they transcend the traditional boundaries, and it's like identifying new questions and new disciplines. So this is a way to view it. But in my case, I think I see it more into like 2 different ways, and it's also continuous. So it's more based on what is the question that the researchers are interested in. So if the question is interesting for one discipline, then you can also work at the interface with other disciplines, where the other disciplines can help you to address the question that you have. And for example, here with our project on the glue, so we are interested in what makes the adhesion different between different species, and trying to understand how this evolves recently. And so here we are working with other researchers working in other disciplines, like in physics or in chemistry and ants. And so here we are asking us to help us on our question, which is our original question, which is really on an evolutionary question. So here it's one possibility and one type of work at the interface between several disciplines. Another possibility is that when the original question is interested not only for one discipline, but also for another discipline. And here different disciplines will come together and try to always reframe the question in a different way, because the two different ways that the question was approached was good, but maybe not as general or not as good as it could be. And then it will be the results that will be found by mixing these different disciplines will be interesting for several disciplines altogether. And I would say that Evo-Divo, I mean, is more this type. So evolutionary biologists and developmental biologists are both interested in the question of how during evolution we went from one type of organisms to another type. So it's interesting for both, and both disciplines are trying to bring their knowledge and their expertise together to try to tackle the question. Of course, I mean, it's not very binary. I mean, it can change. And for example, for our work on the glue, so nowadays we are really working on evolutionary questions, but maybe in the future we will go into a more applied research, try to make a good bioadhesive, and then maybe we will have questions which will all be also of interest for physicists, and then it will be more going into the second part. So when we work at the interface with several disciplines, I mean, it's difficult, but also very interesting. So I mean, personally, I like it a lot. And what is important is not to think that we know everything. We are all biased in the way we view the world and the way we approach research. And of course, the way we were trained biases the way we do research. And we shouldn't think that the way we do it is best compared to what other people do. And of course, the topic we work on has influenced the practices. And so people who work on different objects at different levels will have different approaches and different standards. So it's really important to be open and learn about the new vocabulary used by the collaborators, the way they approach the problem, the methods they use, and the standards they have, because they can be quite different. And about the vocabulary, we have to be aware that the same word can be used by different disciplines, but can have different meanings. And for example, the simple word gene, I mean, we all think, oh, it's simple, but actually there are different meanings. Same for pleiotropy, epistasis, sex. So I will go to this in more detail. So for the concept of gene, with others, I mean, we realize that there are two main concepts. So one is a molecular gene. So it's a stretch of DNA, which is transcribed and which can encode for an RNA or a polypeptide with a known or presumed function. And so this is the definition, for example, in the ModEncode project, or if you look at database of different genes. But then there is also another definition of the gene, which is a Mendelian gene, which is a segregating genetic unit, which is detected through genotypic differences, and which is associated with different alleles at the same locus. So you would think, okay, I mean, nowadays we use mostly the molecular gene, and the Mendelian gene has been lost, but it's not true. I mean, we are using both concepts regularly in different papers. And for example, there was this question on a blog by someone, I mean, how come I share 50% of my DNA with my mother and also 50% of my genes with a banana? And here, the problem is really because there is a mix between the two concepts of gene. So we share 50% of gene with our mother or with our father, because this is when we talk about the Mendelian gene. But then when we say that our genome has 50% of the same gene as with a banana, then we are talking about the molecular gene. So we're not talking about the same entity. So here I put different sentences which I found in different papers where you have the word gene. And this is used in different ways. So for example, here, many of the genes not targeted by our library encode olfactory receptors that are unlikely to be cell essential. So I don't know if you have, you have, no, more. You have reactions, yeah. So maybe, so if you think it's a Mendelian gene, you can upload. Does it work? I don't know if it works. And if you think, reactions, if you think it's a molecular gene, you can put the other one, okay? So for the first one, what do you think it is? And is it a Mendelian gene or a molecular gene? Yeah, Mendelian gene. Okay, so now we will do the second one. So this polycomb repress domain harbor genes encoding key developmental transcription factors whose misexpression can have detrimental consequences in a differentiated cell. So is it a Mendelian gene or a molecular gene? Not so many people replied, but yeah, so it's a molecular gene, okay? And so now the next one, I mean, we will not do all of them. So there has been sufficient time for the corresponding resistant genes to spread into environmental reservoirs. So is it Mendelian? So if you think it's Mendelian, you click on a clapping hand. And if you think it's a molecular gene, you click on the other one. I didn't see anybody, but so this one is a Mendelian gene. Okay, a resistant gene, you know, they can are the genes which are transferred from one generation to the next, and so they can spread from one population to the next. So here we are talking about a Mendelian gene, but these other ones, they were a molecular gene. So I mean, you can do it on the other one, and so this is a thing that we use regularly and even today. So there are different concepts. It's the same for pleiotropy and epistasis. So here I will not go into the detail of this. So what is important when we write papers, when we present talks, or when we have discussions with colleagues which are not exactly in our field, that it's important to avoid jargon and acronyms and try to really precise what we mean. What is also important is to find good collaborators. So usually at the beginning, when we will start working at the interface between several disciplines, we have this problem of understanding each other and also using good and relevant approaches. So it will take time to do good and relevant experiment or relevant research. So it's important to find collaborators which are not into, let's work right away. Sometimes it's really good to think more about what is really the question, what is the best way to address it, and make a good plan before starting and going into a wrong direction. So what I think is much important is really to be attentive to the different colleagues' thoughts, because we think we understand, but actually they probably mean something different. So it's good to really be open and about this and try to understand what is meant. And we all have preconceived ideas, so it's important to be aware that we have them. We don't for sure know what they are, but try to think in a different way. So this is what I like also with working at the interface between several disciplines. So now if we compare microevolution biology with macroevolution, so microevolution is comparing closely related species which can still hybridize in the laboratory, or comparing evolution between population within a species. And so this is most of the work we do in our lab. So we are interested in the microevolutionary mechanisms. And this is different from people who are interested in macroevolutionary patterns. So here across large evolutionary timescales, differences between taxonomic groups and how evolution occurred across millions of years. And so people working on microevolution, usually, I mean, they have a genetic approach or an ecological approach or both, but there are a lot of genetics approaches which are used in microevolution. And usually experiments are done in the lab or in a specific environment. So there are manipulation of the living world which are done, which is different from people who are interested in macroevolution, which are more model-based and computational studies. And usually they compare distant related taxa. So it's more comparative in a comparative framework and using phylogenetic approaches. For microevolution, it will be based on the genotype of the different individuals or comparing different populations within a species. Whereas for people interested in macroevolution, they are focused on different species or different clades. And for microevolution, they will start from the bottom and what is known from these specific differences to try to infer more general patterns. Whereas people interested in macroevolution, they have a more top-down inference. So this, sorry, I forgot to say, this is a slide I took from a very nice presentation by Léonie Moyle that she did online last November. And here I put the link to the YouTube video. And so she explained how she sees the difference between microevolution and macroevolution. So I took this slide also from her. And she says that people working on a macroevolution criticize the people working on a micro, and they say that they are lost in detail, they are down in the weeds. And people working on microevolutionary mechanisms say that the ones working on macroevolution are detached from reality, are up in the clouds. And this is certainly true in some aspects. But what is interesting also to see is that each critic also goes back to his own thing. So maybe people working on macroevolution, they are a bit too much detached from reality, and they don't focus enough on the details, which could also be important. And people working on microevolution, they should try to be more general and be more, a bit detach themselves a bit more from the basic results they found. So I think that it's really good, to try to see the different approaches and try to not see only critics, but see how they also apply to its own disciplines. And so there are gaps between the different approaches because the questions are not always exactly the same. Here the questions about the mechanisms which can explain microevolution patterns versus macroevolution are not exactly the same. So the answers will be different. And so this makes it difficult to combine these two approaches. They're also working on different timescales. The methods, as we have seen, are different. One is more manipulative based and the other one is more inference based, a bit further away from experimental data. And the training and the culture is different. And so there are different biases from both approaches. And so it could be difficult to combine them. So what Léonie Moyle proposed is to find an object which is common to both types of approaches to try to combine them. And she says that DNA and DNA sequence is the same object, and maybe this would be a way to combine it. That's a possibility, but in my case, I think what is important is to try to find common questions that can be addressed by both people working on at these different timescales. And here I want to show you one possibility about trying to combine these two different views. So with this database that we created with David Stern and Arnaud Martin, jiffybase.org, on all the natural and domesticated and experimental variation that has been published. So here we included genes and mutations which have been identified in several research papers, which have been associated with natural phenotypic changes in animals and plants. And we also included domesticated evolution and recently also experimental evolution. But we don't include lab mutants or clinical traits. And so we want to compare these genes and mutations which have been identified across different time scale. And so here, in this paper, which we published in 2009, we surveyed only 300 articles, so about 300 genes in animals and plants. And here we compared the genes and the mutations associated with a short-term evolution, so evolution in domesticated varieties, or also evolution within species. And we compare these mutations with the one which have been identified as differences between closely related species or between species which are in different genus. And so here what you can see is the proportion of mutation. So in black is mutations which are causing which are associated with a morphological variation, and the proportion of mutation which are cis-regulatory. So here there are 60 mutations which are causing morphological variation, which were found in this database. And you can see that about 25% of them are cis-regulatory. And when you look at long-term evolution, you see that the proportion of cis-regulatory mutation increase and is different from short-term evolution. And this is a general pattern, and this, has been confirmed later, including more and more articles. So long-term evolutions involve a subset of genes involved in a short-term evolution, and it's not just a random subset of this gene. It's a specific subset, and the mutations involved in long-term evolution are more likely to be cis-regulatory compared to short-term evolution, which are more likely to be coding mutations. And these coding mutations occurring in short-term evolution, when we look at them, we can see that many of them are null mutations. So null meaning that they remove the function of the gene. So it will be, for example, a stop codon or a change in a frame shift so that the gene is no longer, the protein is no longer produced. And so these null mutation, you can see also the frequency of them is higher in short-term evolution compared to a long-term evolution. And so this to me was a big surprise, because when I started working on microevolution, so comparing closely related species, I was thinking that the mutations that I will find correspond to the one which are also responsible for long-term evolution. And so the patterns that I will identify will also apply for long-term evolution. But actually, it's not so simple. I mean, what we find and what is responsible for short-term evolution might not correspond to the mutations that are stabilized and maintained across long periods of time during evolution. So one example that I can give you is for Arabidopsis thaliana, for example. So nowadays there are many ecotypes which are adapted to different environments, and they flower earlier than the ancestral populations. And these early flowering ecotypes, many of them are due to null mutations in a gene which is called frigida. And so when you have a null mutation in this gene, the ecotypes will flower very early, and this is a very adaptive character. So they spread in many parts of the world. And so this is a null mutation. But this frigida gene is a highly conserved gene, it's found in all plants. So probably over long term, these mutations will not be maintained because they will not survive if the environment change, if the climate change. And in terms of metabolism for the plants, it's better to have a cis-regulatory mutation in the gene, which will affect the gene and affect its expression at a specific time point and in a specific environmental conditions, and not to have a null mutation. So these null mutations are adaptive over short term, but over long-term evolution, they will probably be removed. And then other types of mutation will occur, which are maybe less likely to occur, but with a more specific and targeted effect in the frigida gene or in other genes, which would be cis-regulatory mutation. And so the mutation over long-term evolution are not all the ones a random subset of the ones responsible for short-term evolution. And so this is general. And so now we have more than 1,000 coding mutations curated in a Jiffy-base. And here, if you look at the null coding mutations causing phenotypic variation at different time scale, you can see also that this proportion decrease when we look at long-term evolution compared to short-term evolution. And when you look at the proportion of nonsense synonymous single nucleotide variation in the coding region, we can see also that this proportion decrease based on this database. So this is something that can be fine. So we try to compare the mutations underlying macroevolution compared to microevolution. And it's not the same. And this is an interesting finding. So this is about what I had to tell you about the researcher and his research when it's working at the interface between several disciplines. So now if we look at the other parts of research, so for example, publications. So what matters for publication are two aspects, and this is based on a discussion with Leo Kutelek, who is a philosopher of biology. And so he says that there are two parts. So one part is robustness. So how solid is the data and the results which are presented in the paper. So this is based on a specific methodology, and this methodology has to be accepted by the scientific community. And usually it's based on a conformity to methodological standards, which have been developed by a community in a discipline. So this is the first aspect. And then there is a second aspect, which is relevance, which is how important is the results which have been found for the understanding of biology in general. So this is related to the adaptation of the research to its object of study. I mean, is the question relevant and the way it was done relevant? And does it bring new data which are interesting? So it means that it has to be sensitive to the context and to all the dimensions of the object of study. And also the relationship here to reality and to the real world is really important. If we are working with something which is very far from our world, it will not be as relevant. So these 2 points are important. And when I was working with Drosophila developmental biology, we have a very high robustness because we do experiments and we do them repeatedly, and we always have when we always have the same results, we are really sure about our results, and so we can have very robust conclusion about like this cell divides and give rise to these two cell types, and one will undergo apoptosis and will die, for example. I mean, it's very robust, but then the relevance is not as high as when we are working with Drosophila evolution, because the conclusion here will be about this cell, when it divides, it gives rise to these two cells, and this is specific for a specific system, and it's a bit more difficult to have conclusions which could apply to other systems in general. Whereas for when I started to work on Drosophila evolution and comparing different species, then directly, I mean, even though if it's using Drosophila as a model system, what we find in the field, I mean, is directly relevant to people working on other model systems and to evolution in general. And so here the relevance is higher, I would say, compared to people working in developmental biology. So in a way, we can publish in journals which have a higher impact, because I mean, evolutionary questions are important and interesting for many readers. And so here the relevance, I would say, is higher. But on the other hand, the robustness is not as high, because we are comparing closely related species. We are trying to infer what happened during their evolution, and we will never be sure. It's only hypothesis. I mean, we cannot rewind the tape of life and see what happened before. I mean, so it's just a hypothesis. So we do a lot of experiments. We try to be robust in the way we do our experiments. But I mean, here, I mean, we're trying to infer what occurred in the past. several million years ago, so we will never be totally sure. So the robustness is not as high. And here I put also human evolution. I mean, I have never worked on human evolution, but to me, I mean, my impression is that it's even more relevant. So it can be published in high impact journals. And here, the robustness is not as high as for Drosophila, because of course, I mean, we cannot do as many experiments. We cannot validate our hypothesis as much. So here the robustness is not as high, but the relevance is higher. So we have to combine, I mean, these two aspects, and depending on the disciplines, some will be higher or lower. I mean, here I put them as if it was comparable, but also it can be also sometimes not as comparable. And so in my case, I mean, I was trained in Drosophila developmental biology. So I was used to use a very robust approaches of doing a standardized genetic experiment, doing a lot of transgenesis experiment also to confirm the data. And so I think this gave me an advantage, because I tried to have very solid data, you know, compared to people who were starting with evolution. They are not so much into genetics, and they will be okay with some types of data, but coming from developmental biology, I have maybe a higher standards in terms of robustness. And so this allowed me to shift it a bit this way. And I mean, that's how I see it. I mean, I don't know if it's true, but yeah, so it helps also to move from one discipline to the other, because you can I mean, take things which are really good from one and put it into another one and improve certain aspects of another discipline. So then about the publication, there is a question of in which journal do we publish, especially if the work is at the interface between several disciplines. So what is important when we write the manuscript is that it should be addressed to a general readership. So it should be addressed to several types of readers. So it's really important to define the words, to be very general in the introduction, so that it will attract different types of readers. So there are advantages at working at different disciplines, because sometimes there are journals which are into a specific topic, but they want to expand the research field that they cover. And so then in this, if you are working on this research field, it's easier, I would say, to publish in these journals. So, for example, I was an editor at the Royal at the proceedings of the Royal Society, and so this journal is very, very good in ecology, and you have very good papers. But they want to have more evolutionary genetics papers, and so they would accept evolutionary genetics paper, which would be a bit lower, I would say, compared to ecological papers, just because, you know, they want to increase the range of topics that they publish in their journals. So when you know about such journals, it's an advantage, because it would be a bit easier to publish in them. But then the drawbacks is that sometimes for certain studies, there are no appropriate journals which would fit exactly with the work. So it makes it difficult to have the paper reviewed in a good way and seen in a good way. And also the journal will have difficulties to find appropriate reviewers which can judge your work in the best way. So this can be a problem also sometimes. So there are also issues related to the CNRS. And so, as you know, I mean, in biology at the CNRS, there are several institutes. And for us working in evolution, there are two main ones, INE and INSB. INE, Institut Écologie et Environnement, et INSB, Institut des Sciences Biologiques. So because I was working previously on developmental biology, I mean, and I was, at the CNRS, I applied mainly to INSB section, and so I'm part of INSB, and my institute is also part of INSB. So I have no access to many relevant papers for my research, which are at INE, and I don't have access to them. I mean, it's a bit silly, but it's good that we can use other ways to find papers nowadays. And also, I'm not informed about relevant news and conferences organized by INE, and this is too bad, because it could be really interesting. And I wasn't sure also which section to join because my work is really at the interface between several disciplines. So I thought I would go into organization, expression, and evolution of genomes because it's quite general. But I went into this section for about a year, but I don't know, they changed me and they put me into cellular biology, development, evolution, and development. I think it's because I've worked on evolution, on development for my PhD. So now I'm seen as a developmental biologist, even though nowadays I'm working on the Drosophila glue. I don't do much development. So I think there is a lot of conservatism. I mean, since my master, I think it's better now, but not. It should be still improved. When we are working in one discipline, it's hard to be seen by the others that we shifted and we changed and we are working in new disciplines. And in the end, so I managed to, on the second time, I got on a ATIP Avenir grant on Evo-Divo, which was close to my postdoc work. But the first time I applied to the ATIP, as I told you, I didn't get the grant because it was thought that it was not close enough to my expertise. And I got an ERC grant also on Evo-Divo, which was really close to my postdoc work. But then, project which didn't get funding, so this ATIP grant on Drosophila pacea. And I also applied, for example, to Bettencourt funding to study Drosophila glue. And at that time, I had no paper on the topic, or maybe one only on studying the glue gene sequences. And so it was rejected on the basis that this was not my field and I should not work on this. So I think it's difficult to convince grant reviewers when we are not expert in a domain and when we are mixing several disciplines and when we are starting a new project. So at the beginning, we have to work without specific funding for this and use grants that we have for other projects. But then when there are efforts from the top, from the CNRS, from INSERM or other institutions, then it's possible to get money and a position. And so, for example, my collaborator, François Graner, who is a physicist, he said that at the beginning, when he started to work at the interface between physics and biology, it was very difficult for him. But then the CNRS realized that they had to improve the way, make it easier for researchers to work at the interface. So they created multidisciplinary sections and they created specific grant applications. And so nowadays it's much easier to work at the interface between physics and biology. And this is because a lot of efforts were made from the top to make it easier for researchers. Then there is also the question about the relationship between colleagues. So the main issue is understanding each other. And so I told you already, so we have to learn that we are using the same vocabulary, but maybe meaning different things. And so when we work at the interface between several disciplines, what is nice is that we are used to put our work into a more general and more understandable framework. So this is good when we do, when we give talks or when we write papers. But at the same time, people can have different views on what are the relevant questions, approaches, inferences, and conclusions that can be made. And so it can be difficult also to share our work and to make it relevant for other people. What is also problematic and a challenge is that when we go into a new field, we don't have a reputation. So it can be difficult, especially if we are known in a special field, then we move to a new one. Nobody knows about us, and so this can be difficult. So what can be an approach is to find a common interest, like on a model system or using DNA sequences. And once we have a little bit of a common interest, it might make it easier to interact with colleagues. So I would like to end with one advice that I got from François Jacob. So when I got my position at the CNRS, I was really happy. It was in 2007. And I was extremely happy. So I was happy that he accepted to discuss with me. And so I asked him, what would be your advice to me? I'm just starting at the CNRS. And he said, oh, my advice is that you should have a particular research topic for which you are the specialist. And then I went out of his apartment and I was, oh, what can I do? I mean, it's really not what the advice he could give me, because I like to move and to study one thing and then study something else. So I didn't follow his advice, but I still remember it. And I think it's ideal. I mean, if you have a topic, you are really well known on it and you continue and you follow on it, that's really good. But I couldn't do that. Yeah. So in conclusion, so when we work at the interface between several disciplines, so to me, there are two ways. I mean, one is one discipline is really asking the question and the others are helping. Or it's really combining several disciplines to try to reframe the question and make it good for several disciplines. And so there are multiple challenges doing this work. So challenges for the researcher and his research, but also challenges when we want to publish papers with the research institution to get fundings, and also with the relationship with the colleagues. So thank you for your attention, and I'll be happy to discuss with you about this.